Okay, so why control? Why do I want to talk to you about control when we're at a network neuroscience educational course? So the brain, it constantly must engage control principles. It needs to take stimuli from the environment, process these stimuli, and come out with some kind of behavioral response, which it turns out, I will show you later, that this maps into a target control problem. It must also maintain a tight functional control over the state of its internal variables and transition from state to state, which I will show you as a control energetic problem. So this is the outline of the talk that I'm going to do today. I'm going to tell you what we mean by control in the network context. Control theory has a long and rich history in engineering, and it's only recently that it's been brought to networks. I'm going to explain to you linear control and Kalman's criterion, and then structural control and the concept of driver nodes. Then the individual roles of nodes and links in the network and their contributions to controllability. Then I'll move on to the target control framework and show you the application to C. elegans. And finally, I'll give you a very quick overview of control energy and an application to human DTI networks. So what we mean by controllability in a very broad mathematical sense is if I have some system in an initial state, an arbitrary initial state, can I drive it to some arbitrary final state in finite time? And if we think about something like a car, there are very, very many components connected, and we can have a network to describe how these things are connected. But all we really need is three things to be able to control it. We need the steering wheel, the gas pedal, and the brake, and the clutch if you're European. So when it comes to brain systems, I'm going to talk about dynamics in this sense. We have the whole system, so X is our state vector, which tells us the state of a brain region or neurons. And this is comprised of the adjacency matrix, so the input from other neurons or other brain regions, and then an external input, so stimuli you receive from your environment. Apparently, my next slide has a heavy picture. Oh, there we go. Okay, so this is the linear framework. We're just going to start with this simple equation, x dot equals ax plus bu. And each of the terms in this equation correspond to, so x is the state vector. As I said, this is the dynamics of the nodes, and this can be brain regions or individual neurons. U is the input vector, so this is where you'll apply some external stimuli. A is the adjacency matrix, so this is where we encode the wiring diagram of the system. Again, be this on a macroscopic, mesoscopic, or microscopic scale. And B just tells us where we're going to apply these inputs. It might not necessarily be to the whole system, so this tells us which part to restrict to. If you go ahead and solve this, which is the wonderful thing about looking at the linear case first, you find that the form of x takes this exponential. And if you do a Taylor expansion on this, you can see that x is just a linear combination of the matrices b, ab, a squared, b, etc. So we define this quantity c called the controllability matrix, which comprises this expansion. And it turns out, this is Kalman's controllability criterion, that if the rank of C is n, so if C has full rank, then the system is controllable. And by controllable, I mean every single node in this system, we can drive it to some desired state. And so this comes back to our original problem of from an initial state, we can drive it to any desired final state. So just in this toy example at the bottom, you can see the two input signals U are applied to nodes x1 and x2. And it's encoded in this wiring diagram A, the adjacency matrix. You've been shown the form of this earlier. And B tells us where to apply these signals. We can then perform this expansion and come up with the controllability matrix C on the right. The rank of this, so the number of linearly independent columns, is 4, which is the same as n, has full rank, so the system is fully controllable. So just looking at these little toy networks, are they controllable? I was hoping to include a poll question at this point, but apparently we're not good for Apple Mac. So the first system, I hope is obvious, is controllable. And the second is not. If you were to calculate the ranks of these matrices, you would find that you need a second input signal to the second network to be able to fully control it. So delving into this a little bit more, in real systems, we rarely actually know the link weights. We're more likely to know, does a link exist or doesn't a link exist? Is there a zero or a non-zero quantity? So in this paradigm, we can treat these weights as free parameters, and all we really care about is, are they zero or non-zero? And then we can delve a lot more into these diagrams I was just showing you. So the first big result I'm going to show you is a mapping to a maximum matching algorithm. So just sidestep from control for a moment and consider these networks. A matching of a network is a set of edges which does not share any common vertices. So if you see in the second box, any of these red links, these create matchings because they have nodes that are not in common with the rest. And we talk about a maximum matching when it's one of the largest size. The very top network with 6 nodes, I can't point very well. This is a perfect matching because every single node has one of these matched edges attached to it, right? The bottom ones, there are multiple different forms of this maximum matching. They each have 2 edges, but there's always 2 unmatched nodes. So the green nodes are matched nodes, and the white nodes are unmatched nodes. This crosses over to a directed network very naturally, but now we talk about not having common heads or tails because each connection is directed. So again, we have this example of a perfect mapping with the cycle, and then the bottom network, 3 different ways of coming up with your maximum matching, but in each case there are 3 unmatched nodes and 1 matched node. So the minimum input theorem is that for our system to be controllable, we need to apply input signals to the unmatched nodes. And we're going to call these unmatched nodes driver nodes. They're called driver nodes because they are the ones that will be able to drive the system to the desired final state. So let's have the examples back with these toy networks. So we're going to consider these 3. If you go ahead and perform the maximum matching on it, the very first network, that's just a straight line. You can create a perfect matching in this network. Just not with the first one. You need to apply the input signal to the first one because this node is unmatched. And then you have control over the rest of the network just from this one node. If you have a look at the second network, you create your maximum matching in one of 3 different ways. So that's where we have 3 different sets there. It always comes out with you need 3 driver nodes. It's just not unique what those sets of driver nodes may be. It's just that you will always need 3. So what's really great about this is if we were just considering an arbitrary network and we wanted to find out which nodes would be able to drive it, we would have to do a brute force search with some awful combinatorics of pick 1, then pick all combinations of 2 or combinations of 3 until we show that it's controllable via some driver nodes. This algorithm allows us to do this in polynomial time, which is fast even for large networks. So driver nodes in real networks, what are these like? So in this paper, the authors examine a diverse array of networks, including gene regulatory, social networks, food web networks. There's one neuronal network for C. elegans, World Wide Web. The hypothesis behind this was the gene regulatory network, this would say, have to control the dynamics of a cell, so they might imagine that this would be easy to control. Maybe there would be few driver nodes. And social networks, we like to think they resist control, so maybe we would need a lot of driver nodes for these. This graph shows it better than a table. It's much easier to see. And the results are pretty much opposite to intuition. Gene regulatory networks, they're very hard to control. You need to control a large number of driver nodes. So this ND is the fraction of the whole network, which are driver nodes. So these are the number of nodes, the fraction of nodes that you must input signals to be able to control the whole network. And on the contrary, social networks need very few, which is potentially concerning. The next big result from this is that the number of driver nodes is mainly determined by degree distribution. So what these 2 graphs are showing you is all of these networks we compare to a randomized version of these networks. The graph on the left, the randomized version, is complete randomization, so we're thinking an ER network of the same size. On the right-hand side is degree-preserving randomization, so you'll just perform edge swaps so that each node has exactly the same degree. And we can compare for a random network which has the same degree distribution. And we only lose the number of driver nodes when we do the complete randomization, which is showing us that the number of driver nodes is largely being captured by the nature of the degree distribution. This is good news because this is an easy quantity to calculate for a network. So let's dig into it a little bit more. What do we mean by the degree distribution? There are a few things that could characterize it. When we have a look at randomized networks, as in random, regular, Erdos-Renyi, and scale-free, and we compare the number of driver nodes needed to completely control the network and the average degree, we find that as the degree gets larger, the number of driver nodes goes down. So the denser the network is, the easier it is to control. So these two graphs are now showing you a measure of heterogeneity. This is a quantity that's capturing the average difference between degrees. So heterogeneity is large if you have some very high degree nodes and some very low degree nodes, and heterogeneity is small if they all tend to be a similar degree. And what this is showing is as heterogeneity increases, the number of driver nodes increases. And there's orders of magnitude difference in this. If you get a smaller difference in the nature of the degree between nodes on average, you get quite a large difference in the number of nodes needed to control it. So what's important about this? This means that these two results together are saying that sparse networks and heterogeneous networks are the ones that are hardest to control. And these are the ones that we tend to observe. So particular link and node roles, I'm going to bring you back to this diagram. I already showed you the first two layers. If you now move on to the third layer, we can start to consider the role of every single link within the network. And we can categorize them into three things. One, a critical link, which means that it's always, if you remove this link, the number of driver nodes will change. The redundant link means if you remove this link, then the number of driver nodes will not change. And an ordinary link is one or the other. So in this first case, they're all critical links. If you remove any one of them, it will change the number of driver nodes. You'll have to insert a second input signal. In the middle case, they're all ordinary links because of this multiple way of having 3 driver nodes. And in the final case, we have a mix of all 3. When you have a look at these real networks that I showed you earlier, the fraction that fall into each of these categories, there's a surprisingly few number of critical links in all of these networks. And this is very informative if you want to come to making a network more robust to control, because all you would need to do is pick these critical links and double them up, and then no links are redundant and they're more robust in a control sense. So then to a node sense, we can quantify the amount of controllability that a particular node contributes to the system by considering this thing control centrality. So we already showed that the rank of the controllability matrix tells us the size of the controllable subspace of the system. So it would make sense if we consider for each node I, if we treat that node I as the sole input and ask about the rank of the controllable subspace, then this gives us a quantification of the level of controllability that this node has, right? So this rank G means the generic rank, because this thing depends on the parameters A and B. But for most cases, most values of weights within the system, we arrive at the same rank, and this is this generic rank and the structural controllability that I was telling you about. So it turns out that there's another neat mapping to a graphical solution to this, and I'll just walk you through it now. You need to understand that a state node J is called accessible if there is at least one directed path from an input node to it. So if you have a look at this toy network in the bottom, every node is accessible. If you treat X1 at the top as your input node, you can reach any other node in this network via some path. A stem is a directed path beginning from an input node such that no nodes appear twice. So x1, x5, x7 would be an example of a stem, but it's not the only one. It could also be x1, x5, x6. And a cycle is a directed path which begins and ends on the same node and includes each node only once. So obviously this little circle to the left. Then altogether, a stem cycle disjoint subgraph is a subgraph of G which comprises of stems and cycles which have no nodes in common. So this diagram here, everything highlighted in red, you've got one stem and one cycle, and together they form a stem cycle disjoint subgraph. And it turns out that the rank of the controllability matrix is the same thing as identifying the maximal size of this. So what this thing is saying is the maximum number of edges in your stem cycle disjoint subgraph. And this image contains such a maximal subgraph, and if you count the number of edges in it, you can see that it's 6. So that means that the rank is 6, and that's the size of the controllable subspace. There are 7 nodes, so there's one of them that we couldn't control completely with x1 as the input node, but we can control the state of the other 6. And it turns out that just a couple of little general results on this. The top row, labeled C, if you have an input node coming into any star where things are going out, that middle node in the star, it will always have a control centrality of 2, because all you can do is 1 in, 1 out, and you'll have 2 matched edges. Also, if you have a directed acyclic graph, so just like a tree going downwards, like you're seeing in the bottom, then the control centrality of a particular node I always is equivalent to the layer of the hierarchy that it resides in. So x3, yes, x3 at the top of that green triangle, that has a control centrality of 2. So what do these look like in real networks? Well, it turns out it varies quite a bit. In the interorganization network, so this is like a social network within an organization, within a company, it turns out there's a big peak at control centrality of almost 1. So there's a lot of nodes here which you could control the entire system just from this one node. On the other hand, you have networks like the company ownership one on the bottom right, and this has more of an exponential distribution, which says that there's a few of these that could control the whole system with just from one input node, but a whole bunch that you would need lots of them. And then other networks tend to have a bit of a mix of both, where you have some nodes with a high control influence and some with a low. And similarly to what we observed earlier, when you compare these to randomizations, if you can see on the little inserts, we find that the degree distribution, again, is behind a lot of this control centrality. We only lose the results when we go to an ER graph, and we maintain them with the degree distribution. Okay, so I've been talking about controlling the state of every single node in a system, but we often don't want to do that. We often only want to control a subset of these nodes. So this is where we start to talk about target controllability. So I already told you the top line in this equation, and we're just going to add in one more. And this is going to be this y of t is cx of t. So this is going to be the dynamics of the output nodes, whatever things we might be interested in. And that's going to be described by c, which tells us which nodes we're interested in, and then x of t, the state of the system. So A, B, and C, this is what we were just talking about before, where we would have to identify any one of the 3 nodes with the input arrows to them. Our maximum matching theorem tells us that we would need to control those 3 to be able to control the whole system. But if we only care about controlling the output nodes, the green ones, if we call these the target nodes, then D tells us that we only actually need 1 input signal. So this is the K-walk theory, where all we are trying to say is that these paths are of unique length. So the path to 1 is 0. This is where our input signal is. The path to 2 is of length 1. The path to 5 is of length 2. And the path to 7 is of length 3. So because these are all different lengths, this means we only need one input signal to drive all of them. This stems from the fact that we're really looking for linearly independent signals. Okay, so we can solve it with the K-walk theory with just one input. But if we want to go to more, we need to consider something like a greedy algorithm. So in iteration 1, all we're doing is performing a maximum matching like we did before, but we've created a bipartite graph kind of by stretching out one in time. So if you see the connections in this bipartite graph are just the ones that exist in the normal graph. We perform a maximum matching on this, and then we take the nodes that are matched, and we use those as start nodes for the next iteration, and continue until we arrive in iteration 4, where we see we only need this one, the driver node 1, to be able to control the whole network, so the particular output nodes. Again, more generalizing this, if we have many inputs, there's only one in this picture, but this generalizes to many, we can construct this thing called a link graph, which is essentially taking those bipartite networks I just showed you and stretching them out. And if we solve the maximum flow problem on this graph, we can come out with the maximal number of these output nodes that we can control. So back to these toy diagrams, you've already known from what I showed you earlier that the first diagram is uncontrollable. The two pink nodes would have to move in some correlated sense, and the second one is controllable with the second input signal. But now we can start to talk about how controllable. So if we consider row C, if you have all of these signals coming in, if there are m of them, if m is less than k, then we could control all of the nodes in the bottom layer. We could control m of the nodes in the bottom layer independently. The rest would have to be correlated. So an application of this to C. elegans. This is the C. elegans response to gentle touch. C. elegans moves in sinusoidal waves. If you touch it on the front or the tail, it will perform a reversal and crawl away from you. And you can frame this in a target control sense. There's an input being received by these mechanosensory neurons. This is processed by the nervous system, and it's output to the muscles in this measurable behavior. So this is what the C. elegans connect home looks like. And it's a dense mess is the point of this picture. And if you'll probably have to squint to see, the input nodes are buried somewhere in the middle by the big orange nodes. And the control framework is going to help us tease apart the role of these individual neurons. So what we're going to do with this framework is set it up like I told you with your input, your system, and your output. And then for each node in the system, you systematically remove this node and ask yourself how many controllable output nodes, which is in this case the muscles, do I have? If this is different than the healthy level of controllability, then this node matters for the behavioral response. If it is not different, then it does not matter for the behavioral response. So if you do this and you work through all 279 non-pharyngeal neurons, you only come out with 20 that are predicted to affect muscle control. The one I'm going to focus on, because time is short to show you, is the DD neurons. So there are 6 of these motor neurons, and they were largely tested together as a class. They're morphologically similar, they have similar connectivity, and they were all thought to be involved in locomotion. But the control framework implies that only 3 of them, DD 4, 5, and 6, are essential for locomotion, and DD 1, 2, and 3 are not. So what's actually going on in the network here? It's a bit of a messy network, but the thing that you need to see is the ones in the green boxes, DD 1, 2, 3. If you knock any one of those 3 out, it doesn't change anything about the controllability of the muscles below them. But the ones in the red boxes, if you knock one of those out, you lose controllability over one muscle, so there's more correlation in their movement. So what does this look like if you go ahead and actually do the ablation? So this is how C. elegans normally moves, just in sinusoidal waves. And if you ablate one of the predictions, DD5, it starts dragging its tail behind it. Cool. So now we're going to move on and give you a brief overview of control energy. So what I mean here is that there is an energy landscape that the brain must transition between, and the idea behind this kind of approach is that some trajectories will be easy to do and some trajectories would be hard. So you might find that the brain favors to go in particular directions and avoids others because it's prohibitive in cost. So in general, we can describe energy as a function of the input signal as an integral of this, and the minimum energy required to move from some initial state x equals 0 to some desired final state xd is given by this equation, which centers on this object called the controllability gradient. This depends on the final state and on the time required to get there. So here's a toy example of what's going on. We just have this little 3-node network with one input signal in the top. The negative self-loops mean that the eigenvalues of the matrix are negative, which is unusual and makes this harder. And then we pick 3 desired final states. And in B, you see the optimal input signals which drive it to those final states. Each of these final states is only a distance of the size 1 in the state space away, but they take very different routes to get there. If you have a look at C, you see the very small trajectory through state space compared to the very large one. And D shows you the surface of the energy. So it's got the eigenvectors of the controllability gradient define this ellipsoid, and this tells you how much energy is required to move in different directions in the state space. And the point of this is that it varies in massive orders of magnitude. So you can have some directions that are very easy to get to and some that are incredibly difficult. So again, in the context of the brain, you find that there'll be some states that are accessible, some that are too costly, and perhaps some that are even damaging. And when it comes to talk about health and disease, we might talk about how we want to move the brain from one state to another. So in application to human DTI, some more work from Danny Bassett's group, they adopted discrete linear time invariant dynamics. So all that's different from what I was showing you before is this is discretized. They consider DTI networks from 8 individual subjects. And they first ask, is global controllability possible? Is it possible to drive the brain to a desired final state just through one region? And they find that the eigenvalues are very small but positive. So that what this means is, yes, it is possible, but it's very, very difficult. So they dig into this a little bit more to try and understand the role of particular regions. So without delving into the details due to time, average controllability, what they're trying to capture here is the average influence of controllability over all different target states. So you consider, if you had this region as a control node, the amount of energy it would cost you to move to different final states and average over all of these, and this gives you some measure that captures its average influence over easy-to-reach states. The modal controllability is trying to capture the ability to drive to hard-to-reach states. So these things that are very far away, if a region is capable of driving there, then it may have a high modal controllability. And then finally, boundary controllability. So what this does is consider community partitions of the network, consider the nodes at the edges of these communities, and if you are nearer the boundary and more capable to move across communities, couple trajectories or decouple trajectories of these disjoint communities, you'll have a high boundary controllability. So what does this look like in their DTI networks? They find that average controllability correlates very highly with weighted degree. So if you have a high degree, you have a high average controllability. So this will be stuff like your rich club nodes that you were told about earlier. They make it easy to move to easy-to-reach states. Modal controllability, this has a negative correlation, so your low degree nodes are the ones that have the high modal controllability. These are the ones that help you reach the harder-to-reach states. And then boundary controllability doesn't really have any discernible relation with degree. So where do these sit? We can decompose the brain into a number of systems, including the default mode network. And this is where we find there's a high average controllability, but a reasonably low modal and low boundary controllability. And then they find that the strong modal controllability appears in cognitive control networks such as the frontoparietal and I can't pronounce the word, opercular. And regions of the attentional control systems form strong boundary controllability hubs. So these are the ones where you might synchronize states more easily. And then this is just going on from the same thing. You find the hubs, so they consider the top 30% of control hubs. And the hubs of average controllability are preferentially located in the default mode network, which agrees with the previous graph. And hubs of modal controllability are predominantly located in cognitive control networks. And hubs of boundary controllability distributed through all systems, with two predominant systems being ventral and dorsal attention systems. And finally, they find that there's a tight relationship between development and these two types of controllability, average and modal. It seems that the human brain develops to optimize both of these things. It is important to have a high average controllability so that you can flick between these states, but also a high modal controllability. And at some point during development, there are changes that move towards this optimization. So we can use controllability to start to dig into regional variations and the principles underlying the way the brain is controlling itself. Control is essential to any neural system, as I said at the beginning. It must be able to continuously maintain a tight functional control between its inputs and outputs. I've quickly gone over some of the fundamental concepts of network control, including Kalman's conditions, structural control, driver nodes, control energy, and you've seen a couple of quick applications to C. elegans into human. There's a lot I've not managed to cover, and I hope I've done a little bit of justice to just skimming over the surface of these, and just that this inspires you to look out the rest. So there's some resources for you. So some of the code for a lot of the things I said you can find on Yang Yu's site. My GitHub page for C. elegans control and Danny Bassett's page has got some data sets and some of the control energetic stuff I think you would have to request. I also instruct a graduate class at Northeastern, and we have a lot of materials online for just network science in general, if any of you are interested. And we teach a lot from Laszlo Barabasi's book. And then finally, I want to recommend this book by Stephen Schiff, Neural Control Engineering, which looks at the really neural side of control as opposed to brain regions side. So thank you, and I hope I've got a couple minutes for Q&A.